So the fact that there are no obvious environmental inputs in this azenic culture for generating a heterogeneity of the population suggests that instead purely stochastic factors, random fluctuation from cell to cell in gene expression, interdivision time, and so on, could play an important role in generating the persister phenomenon. So we would like to postulate that stochastic processes may in fact underlie the fundamental phenotypic switch to the drug-tolerant persister state. Now, this does not, of course, preclude a role for environmental factors. In fact, environmental inputs could have a modulatory effect or a stimulatory effect on the rate of switching, even if the underlying switch mechanism is essentially stochastic. And there are excellent examples of this from model organisms that have been well studied, probably the best studied of which is the switch that occurs at the lac operon in Escherichia coli. So I'm sure all of you have studied the lac operon at one time or another in your undergraduate studies. I'll just remind you of the components of the system. The lac operon encodes a beta-lactamase, coded by the LAC-Z gene, a permease, LAC-Y, that brings lactose into the cell, and the beta-galactosidase can then act on that lactose that's brought into the cell to convert it in part to allolactose, which is the true inducer of the system. Allolactose then binds to the LAC-I repressor and causes it to fall off the DNA, thereby de-repressing expression of the lac operon. So again, the sequence of events is lactose enters through the LAC-Y permease. That lactose is then converted to allolactose by beta-galactosidase. That then causes de-repression by binding to the LAC-I repressor and causing it to fall off the DNA, leading to de-repression of the operon. So it's a very simple, lovely architecture, but it obviously can't work, as I've drawn it here, because in an uninduced cell where the lac operon is repressed, there's no LAC-Y. There's no permease to bring the lactose into the cell. And even if the lactose could enter through, say, simple diffusion, there's no beta-galactosidase to convert that lactose to the true inducer, which is allolactose. The reason why this system works is that, in fact, it's not perfect. It's leaky. In a subset of cells, there's always some expression of the lac operon, and this is simply because repression depends on the continued interaction of the LAC-I protein and the LAC-O operator on the DNA. And like any intermolecular interaction, it breathes a bit. Sometimes the LAC-I just falls off randomly, and when this happens, there's some transcription of the lac operon leading to some production of LAC-Z and LAC-Y, and that subset of cells in which these events have occurred are now primed to respond if lactose happens to be added to the culture at this time. So there's always a subset of cells, even in a clonally homogeneous population, a genetically homogeneous population, that are primed, if you like, purely by accident to respond to lactose if it should happen to enter the environment. The key, as I said, is LAC-I and the interaction of LAC-I with its operator. Now, it's important to note that although this is essentially a stochastic switch that occurs when LAC-I happens to fall off the operator, the rate at which actual induction can occur in switching to the LAC-on state is modulated by environmental factors, most notably by the presence of other sugars like glucose in the environment. Glucose is a preferred sugar over lactose, so when that is present in the environment, even with addition of lactose, the rate of switching is modulated because glucose blocks the uptake of lactose, and it also represses the CRP transcription factor, which is required for expression of the lac operon. So here we have an example of what is fundamentally a stochastic switch to a state that's primed for responsiveness to lactose, but where that switch, that phenotypic switch, can be modulated by the presence of other environmental factors like glucose. What this kind of architecture then leads to is a bistability of the response of the bacterial population to an inducer like lactose. When lactose is added, those cells that are primed to respond completely and rapidly, and they induce the lac operon to its full state. In those cells in which LAC-Z and LAC-Y are still absent, when lactose is added, they're essentially blind to it because they can't take up the lactose and they can't convert it to allolactose, and so they stay in the uninduced state until, by random chance, LAC-I falls off of its operator and allows those cells to become primed. And because this is essentially a positive feedback loop, once cells have entered a state in which they're capable of being induced, they are very rapidly induced to the full state. So in a situation like this, as shown graphically here, if you look at a population of cells after addition of an inducer like lactose, where the cells have been engineered to express GFP once the lac operon is turned on, it's the green fluorescent protein, a marker that can be used on a single cell basis, what you find is that the population of induced cells consists of two discrete subpopulations. These white cells are cells that were not primed to respond to lactose at the time this inducer was added, and therefore they have not. So the lac operon is still off in these cells. In contrast, these green cells were primed to respond, and they have by completely inducing the lac operon. There's no intermediate state. So in fact, cells are on or they are off. They are not partially on or off. So this is a bistable phenomenon, rather reminiscent perhaps of the persister switch leading to a subpopulation of cells that are refractory to chemotherapy, although in that case we do not understand the underlying mechanisms.